Hello everybody, welcome to this webinar that will show you the feature viewer recently launched by UniProt in the UniProt website. My name is Leila Garcia. I'm a software engineer in the UniProt team at EBI, and I'm going to show you today the feature viewer and how it can be used. Okay, so let's going to start with just a brief introduction about the UniProt, which is the Universal Protein Resource. We aim to provide very high quality and very comprehensive data related to protein and protein annotations. So we provide detailed information about the protein function, also interactions and pathways and everything that is related to the protein. We also provide the sequences, and we include isoforms, disease variants, and post-translational modifications. And we always provide stable identifiers that are called accessions. So at any time, you can retrieve the information about the protein you are interested in. UniProt is a consortium with members in 3 different countries. So we have PIR in the United States, we have SIP in Switzerland, and EBI, which is located in the United Kingdom. Let's go to start now with what we want to present to you today, which is the feature viewer. So as you probably know already, UniProt is offering a large amount of protein sequence annotations. These sequence annotations describe regions or sites of biological interest. For instance, we have secondary structures, we have domains, we have post-translational modifications, binding sites, active sites, or transmembranes, and some others. And all of them play a critical role in the understanding of what the protein does and what the protein function is. The sequence annotations in UniProt are distributed along the whole entry, so you can find these tables showing the sequence annotations in UniProt in the different sections, and all of them are in a table showing the feature type, the position where the feature is found, the length, the description, and there is like a graphical view that just localizes the feature within the sequence. And they can be as a very long table as well, separated from the rest of the information in the entry if you go to the feature table on the left side of the entry. You need to be already in a UniProt entry in order to see all this information. So the tables are useful, but there are so many annotations, and sometimes it can be difficult to get all the idea about what all these sequence annotations are telling us and what is the information that they are providing. A graphical compact display would be useful in this case because the visualization of all these annotations in the same place may allow users to discover important biological information. For instance, if you are looking for a variant which is deleterious, meaning that it will damage something related to this variant, and it is colocalized with an important protein motif or an alpha helix or an active site, for instance, then that might be a disruption of the protein function. That is something that you can get more easily with a visual display than with a table-based display. So that is the motivation that we have for the feature viewer. You might have heard this sentence, if a picture is worth 1,000 words. Well, we think that that is true, but we also think that with interactive visualizations, we are offering more than you can offer with just teras of data just in a raw format. So we have now this visual display, and we actually have it since January this year, since the first release this year of UniProt. And it is available for all UniProt entries. If you are already in a protein entry, you will see on the display section on your left this feature viewer link. So all you have to do is just click on the link, and then you will get the feature viewer, which will look as what the slide is showing us right now. So now we are going to see the different characteristics that we have in this feature viewer, and then you will learn how to use it and the different things that it is offering. Let's start with the ruler, which is located on the top. It is giving you all the information related to the length of the protein. And it has anchors on the left and on the right that will make you easier for you to zoom in and to move to a particular region within the sequence protein. In this slide, we have already used these anchors in order to zoom in to a smaller region than the whole length of the protein. There is a different way to do a zoom in, and is if you localize your mouse in the middle of the feature viewer, then you can just use the middle wheel, and then you will zoom in or will zoom out, depending if you are moving the middle wheel to up or bottom. You can also use buttons, and we have 4 different buttons. If you are looking right now on the website, you are probably missing the information button on the very left, but that will be added soon in about 2 weeks. So this information button will take you to the documentation that is available in GitHub. It is in GitHub because our feature viewer is an open source JavaScript component. We will go back later to this point. We also have other 3 buttons that will make it easier for you to zoom in directly to a particular point within the protein. We also have a button that will allow you to reset the view. So if you have zoom, if you have open things, and you use this reset button, then you just will go back to the initial state. And the settings buttons that will allow you to hide or show things. So let's go into see now how to select a feature. In order to select a feature, you just have to click on it, as simple as that. And as soon as you do that, a tooltip will be open with more information. The information that it is displayed in this tooltip, of course, depends on the information that it is available about this feature in the UniProt database. So for some of them, we will have more information than for some others. The information is the same that we are displaying within the table in the entry. You can get rid of this tooltip at any time by clicking on the X or just clicking somewhere else in the feature viewer. The tooltip just will go away. You can select another feature at any other time. Only one feature can be selected at a time. So as soon as you select, click on other feature, then this new feature will be selected and the other one will be automatically deselected. Please note that we have some special features that are not covering a whole region, but just covering 2 points, for instance, like the disulfide bonds, or just covering typically 1 amino acid like the binding sites or the catalytic sites. So those are represented not as a region, but with different shapes. For the disulfide bonds, we use bridges, while for the others, we have created a specific shape for each one of them. Now let's go back to the zoom with the buttons. So at the beginning, you will have the zoom button with the plus. If you have selected a feature and you click on this zoom plus button, then you will be zoomed directly to the region that it is corresponding to this feature. And the selected feature will be exactly in the middle. And because you have zoom in, then the button will change to zoom out. You can reset to the initial state at any point with the reset button, and then you just will go back to the initial feature viewer display. You can also hide and show categories. Let's suppose that you are not very interested in molecular processing, so you don't want to display these features. You can go to the settings buttons, and then you can select which categories you want to display. You can use it at any time to show or hide any category. The categories, which are the ones that are on the left, are initially closed, but they can be expanded. And when they're expanded, then you will see tracks that will show you each different feature type in a different row. So you will realize easily about what each shape is about, or where are all the disulfide bonds, or where are all the glycosylation sites. We have a special track, which is the variance track. This one follows a different visualization from the rest of the tracks. As you can see in this slide, we use a matrix-based view for the variance. In this matrix, the y-axis contains all the 22 amino acids plus a star. The star is used to represent stop gain and stop loss and init codons within the sequence. And the X scale, as in the rest of the feature viewer, remains the position alongside the protein sequence for the feature. For the variance track, we also provide some filters, so you can easily distinguish which variants are related to diseases or which ones are not, which ones are provided directly by UniProt and have been curated by UniProt curators, or come from large-scale studies. These large-scale studies are, for instance, COSMIC 1000 genomes, ESP or EXAC, and we are importing information from all of them in order to display a very comprehensive view of the variants within a protein. You can also get the information about these variants as an FTP from the FTP UniProt website if you want it like in a file rather than in a visual display. We also use a variant color scale, so you can, based on the color, distinguish which variants are deleterious and which variants are benign. So our scale goes from green to red. The lightest green will be non-disease associated. The darkest red, which is like a burgundy, will be disease created by UniProt, and anything in between will correspond to the larger scale studies from benign to deleterious. We have a special color for the init codon, the stop loss, and the stop gain, which are located in the very last row of the variants with the star that we already talked about it, and that color used for them is blue. For the variant tooltip, this one will be bigger than for the rest of the features because we are providing more information that comes from UniProt, but also information that comes from the larger scale studies. And then you can see everything related to the information. Something that it is important here is that in this tooltip, the information that comes from UniProt is the information that you can see also in the UniProt feature tables. But the information that comes from the larger scale studies is only available via FTP or in our feature viewer. That one you will not see it in the entry. So this is the way that you can use the feature viewer, and what we aim is to make it easier for the UniProt users to see in a compact view what is happening with all the sequence annotations. And as we mentioned at the beginning, with this display, you could easily spot what features are collocated and then getting an idea about what is happening in that region of that feature that you are interested in. Let's go in to move now to the implementation. It's not magic. It's a team of 3 people working on this feature viewer. And as I already mentioned, it was implemented in JavaScript. It uses D3 as the main library for the visualization, but it is also developed as a Bio.js component. Bio.js is a community-based project that offers a registry with different components that mainly work with biological data, either for visualization or for parsing these biological data. So the aims of Bio.js is making modular components that are easily reusable and extensible. So that's why we decided to use the Bio.js guidelines to develop JavaScript components. The code is available. You can find the code in GitHub, so you can download the code. But you can also use the code via NPM. NPM is a registry or a repository that provides different JavaScript components that have been created with a technology which is called Node. There you can find the component, and then you can download it and you can install it, so you can actually use it in your own website if you decide to do so. You will find all the documentation in GitHub about how to install the component, but also about how to use it. And all the data that we are using for this feature viewer is provided by the new feature data services by UniProt. EBI, it's a RESTful interface that will provide information in JSON, but also in XML. In this slide, you have the link that will take you to the documentation. These web services are not officially published yet, but they will be for our 5th release this year that will happen in the 2nd week of May. Then they will be publicly available and they will be officially released. And as I mentioned, you can get the documentation in GitHub for end users, but also for developers. And that has been our presentation for today about the feature viewer. So please let us know if you have any questions, and we will do our best to answer. Thank you very much.